Hey, Mrs. Gabriel here with another Gabriel interpretation. This one is on cell signaling and bringing it all together. Let's see if we can get this to work. All right. In order for multicellular organisms to live and flourish, they must perform communication between cells. Of course, the three stages of cell signaling, which are absolutely essential to know for the test, are reception, transduction, and response. In reception, a ligand, which is a signaling molecule, is detected by the target cell. It usually will dock on the outside, as shown in this diagram to the bottom, but there are other means of doing this, which we'll discuss later. So step one is reception. The signal is received. Step two is transduction, a step or series of steps that will initiate a specific response from the cell, sometimes called a signal transduction pathway. Let's be honest, this is usually a large biochemical pathway that occurs. Step three is the response. Any number of cellular activities can be the response. You could create a protein, increase cell permeability, rearrange the cytoskeleton, turn on a gene. It's practically an endless array of what can occur. So, reception, transduction, and response. So signaling has two main varieties. We refer to them as localized signaling and long-distance signaling. In localized signaling, it takes on a lot of different forms. There are gap junctions or plasmodesmata, where there are connections between the cytoplasm. There are surface-to-surface ligand receptor binding, that's one type. Paracrine signaling with cytokines, autocrine signaling, which is very strange. And we'll look at some pictures here on the next slide. Long distance signaling, it covers a long distance, usually moving molecules through the bloodstream or a plant's vascular material. Sometimes it can even be through the air, which we'll discuss later. Endocrine signaling and neurological signaling and gases in plants are your biggest examples. Kind of wondering if immunity couldn't work here in some ways, but they do usually touch one another, so that might be really considered more localized. So here are some of the pictures. It's not the darkest. The top one, you have a gap junction. The second one, you have contact-dependent signaling. The third one, you have paracrine signaling. So a molecule is released by one cell, and it initiates a response in the surrounding cells, but it doesn't go a long distance. Strangest one to me is autocrine signaling. So the cell secretes a type of signaling molecule that goes to the outside and bonds with the receptor on its own cell membrane. Kind of weird. Then you have endocrine signaling there, where you have some type of an endocrine gland that has secreted a signaling molecule. It travels through the bloodstream to the target cell and initiates a response. And there at the bottom, then we have electrical signaling via chemical transmission. There's a couple different ones that we could think of here, and that is with one neuron to the next or one neuron to a muscle cell. So what are signaling molecules? Signaling molecules can be a variety of things. There are steroid-based signaling molecules and peptide-based, which we know is a protein. So the steroid-based signaling molecules are usually derived from cholesterol and considered to be steroid hormones. They are hydrophobic, and they need to be carried through the bloodstream but move directly into the cell to a receptor that's located within the cytoplasm. They are slow to initiate, but they have long-lasting effects. The second one, or the other type, is the peptide-based, and they are proteins. They can be large or small. They are water-soluble, and they cannot move directly through the plasma membrane. They must dock at a transmembrane receptor or an intermembrane protein. A transmembrane receptor is just a type of an integral protein. These do produce a much faster response, though. So here's two different ways of looking at it. Our first one is the way that a peptide hormone would be received on the outside of a cell. You would have its reception out there. We would have transduction with a lot of different chemical reactions taking place, and then some type of response. With this hormone here that is in the portrait orientation, we have the hormone aldosterone, which can go right through that phospholipid bilayer there, and it can be received within the cytoplasm. It is usually going to act as some type of a transcription factor to initiate a cellular response in the form of turning on a gene and creating a protein. So let's look at some of the transmembrane proteins. So several transmembrane proteins exist on each cell, and we have a variety of them. They act as docking stations for a ligand, remember, which is just a signaling chemical, or they can act as a receptor to detect foreign particles, as within the immune system. As ligands encounter cells, they only react with cells that have receptors for that particular signaling molecule. It's very important to remember that. The same ligand can elicit different responses in different cells and tissues. So, epitopes are regions of amino acids on transmembrane proteins that may be seen as foreign to the body. Epitopes elicit an immune response, and we'll talk a little bit about them later. So one of the types of transmembrane protein receptors is called receptor tyrosine kinase. Sometimes they're called RTKs. They have an enzymatic activity that catalyzes the transfer of a phosphate from one ATP to a tyrosine on a substrate protein. This is going to activate a lot of relay proteins to begin a signal transduction pathway. Kinase, think of kinetic energy. You are going to energize this and you're going to put it on the move. So ligand-gated ion channels, we're going to talk about these a couple different ways. They help determine if a certain chemical can enter the cell at any given time. They're incredibly important in action potential in a neuron or in a muscle. So one of the strangest ones is one called a toll-like receptor. It's found in the membranes of innate immune system cells, such as neutrophils and macrophages. And they have the ability to recognize non-vertebrate molecular patterns such as double-stranded RNA and lipopolysaccharides that we would not have within our own body. Toll-like receptors, or TLRs, basically find foreign particles and bind to them. So some may internalize the foreign particles and initiate an endosome, which is basically like creating a vesicle. That vesicle will then merge with a lysosome to break down the foreign particles. So another one is a G protein coupled receptor, which activates when the ligand is received, it activates the GTP and then initiates a cellular response. Now, let's do some specific examples that are specific to us. Here we go. Neurons and action potentials. So CNS versus PNS, central nervous system versus peripheral nervous system. The central nervous system is your brain and spinal cord, and they are amitotic cells. Your peripheral nervous system is all of the nerves, the things that we think of as nerves, which are truly neurons, and they lie outside of the brain and spinal cord. These are both our motor neurons and our sensory neurons. So membrane potential. So the membrane has a specific type of an electrical current, and the membrane potential is maintained by a lot of transmembrane proteins that are going to regulate things like sodium, potassium, and chloride ions within the cell. The transmembrane proteins that are important in this could be potassium leak channels, ligand-gated ion channels, sodium-potassium pump, and voltage-gated channels. So those are a few of the membranes. If you look here, we are looking at a basic generic cartoon diagram of a neuron where we have the dendrites that will receive the information. You have the cell body with the nucleus there. You have this area that funnels down into the axon, which is the axon hillock. That is where the decision is going to be made if we are going to send the signal forward or not. We have Schwann cells, which are myelin sheaths that we'll talk a little bit about later. And we have the axon terminals, which are going to turn the electrical signal into a chemical signal so that it can go into a synapse and communicate with the next cell. So let's talk about the action potential. What is an action potential? It's really stimulating a cell to move and to send a signal forward. And action potentials are only possible in neurons and in skeletal muscle cells. So when a neuron is totally resting and it is not being stimulated anyway, it's said to be polarized. So when some signals start coming in through those little dendrites, if you have enough signal coming in that, hey, I really need to respond to this, you're going to accumulate enough signal to reach what we call the threshold potential. So as the cell is in a resting polarized state, its voltage it lies at around negative 70 millivolts. If you are receiving a little bit of input, if you can reach up to a threshold of negative 55, from that point on, you have initiated the beginning of an action potential where you're going to have your sodium ions are going to start to rush into the cell. Potassium ions are going to leave. You are going to flip the electric current within the cell, and depolarization is going to occur. Depolarization results in an action potential that is at the apex or the tip. And then after you have reached your action potential, your sodium, potassium pumps are going to kick in, and they are going to try to restore the membrane potential. What happens though is because these cells also have potassium leak channels, which are always open, we're going to basically overdo it. So we're going to enter hyperpolarization for just a brief moment, and then everything, as the leak channels remain open, can balance itself back out. And this period is also known as a refractory period. For that small moment, that cell cannot be restimulated. Okay, so let's talk about sending the signal forward. First of all, if you remember, there's the little dendrites on that picture right there, and we're trying to show the axon terminals encountering the dendrites of the next cell. So the cell that this is occurring in has a cell in front of it and a cell behind it, and we call the one the presynaptic neuron and the next one the postsynaptic neuron. So as we get to the end of that presynaptic neuron, that electrical signal is going to have to be turned into a chemical signal where it will release neurotransmitters into the synaptic cleft, which is basically a little trough in between the two cells. The neurotransmitter that's generally spoken about is acetylcholine. Acetylcholine will then bind to the ligated ion channels, and then we're going to have a lot of interesting things happen, including sodium start to rush into the cell, potassium starting to rush out, and we're going to go through an action potential of depolarization and then repolarization again. But how do we know if it's going to send that signal forward or not? We talked about there needs to be enough information coming in from the dendrites to actually overcome threshold potential. Well, all of your signals coming in are not all going to say, yes, push through, push through. Some of these signals are going to be inhibitory. So inhibitory postsynaptic signaling and excitatory postsynaptic signaling, those are IPSPs and EPSPs. So it's basically kind of like a tug of war in whichever one has the most signaling molecules come in, it's either going to send that signal forward or it's going to just die out. So we spoke for a moment about the myelin sheath along the axon terminal. There's some adaptive advantages with this. And if myelin is present on the axons, the signal can be sent faster. It's like insulating a wire. This increases speed and rate of conduction and rate of depolarization of the cell. There can also be some problems with this. If you have cells that need to be myelinated and you have a mutation within a gene, it can really, really be a developmental large problem where cells do not act as they should. So let's look at stimulating a muscle to contract. This is considered to be, and so is the communication from one neuron to the next. It's considered to be a localized event. Even though we're moving a long distance, it is two cells in close contact that are signaling to one another. So this is a picture of an axon terminal of a nerve cell that is reaching a muscle cell. So let's go over a couple terminology terms here. So our first one is the sarcolemma. If you look at the blue-purple line, the blue-purple line is representing the cell membrane, in this case called the sarcolemma. Another important organelle within a muscle cell is called the sarcoplasmic reticulum. It is basically the endoplasmic reticulum. It is incredibly important in maintaining the calcium within the cell, and you will see why that's important in a minute. We also have a chemical compound referred to as actin and myosin. Now, actin and myosin are so plentiful that they make up these bundles called myofibrils within the cell. But what are actin and myosin? They're contractile proteins. They're basically components of the cytoskeleton, and they need to be able to slide past each other and bind to one another to create a cell contraction. So as you can see here at the end of the axon terminal, it's going to release acetylcholine just like it did in the last picture. It is going to bond to some ligand-gated ion channels, which are not shown directly here. That is going to cause an action potential along the entire sarcolemma, and it is going to initiate the sarcoplasmic reticulum to release calcium ions. So let's take a closer look at this. So here you have at the very top, you have the axon terminal. It is in conjunction with the sarcolemma. It's showing here the sarcolemma. This one shows actual ligand-gated ion channels and excitation occurring. Excitation is basically the action potential which runs along the sarcolemma. Interestingly, the sarcolemma has these little divots called T-tubules, which allow that electrical impulse to travel deeper within the cell itself, which is going to initiate this brown thing that looks like an ice cream scoop to me, and that would be our sarcoplasmic reticulum. It's going to release its calcium ions. The calcium ions act as a ligand, which are going to go over and they're going to bond with a troponin molecule. I kind of refer to the troponin as a repressor protein because it will not let the actin and myosin touch one another, so they are repressing the situation. Once that calcium binds, the troponin is going to shift its position and release a binding site where the thick myosin, which is purple in this diagram, is able to now make contact with the thinner green actin filament. This forms what we call a cross bridge, and this is going to cause muscle contraction. So that is a really good diagram that we just looked at. So another type of signaling that we're going to look at, this one is actually more of a long distance one. It is bone remodeling. So calcium regulation within the body is often referred to as bone remodeling, and we just saw how important calcium is. So we need to make sure that your body is supplied with calcium on a regular basis, and if it is not, some bad things are going to happen. So this is a type of endocrine signaling. And in endocrine signaling, that means that there is going to be a signaling molecule that is released from an organ, and it is going to travel through the bloodstream, and it's going to initiate a particular cellular response. So let's talk about these terms. What you're seeing here in the top right is actually real blood. It is real bone. It is compact bone, and in the dark circles in the center, that's actually the blood vessels, along with some nerves that run through that. That is a Haversian canal or a central canal, which holds the bloodstream. If you look, it looks like rings of a tree. Each of the dark dots is going to be an actual bone cell. Everything that looks white is what we refer to as bone matrix, or bone to you and I. It's full of calcium and phosphorus salts. So how do we regulate blood calcium levels? Well, we either withdraw it from our bones or deposit it into our bones. So let's go through a scenario. When calcium levels drop, PTH is released by the parathyroid gland. PTH is parathyroid hormone. Osteoclasts, and you see the word osteoclasts there, they're going to have the ability to collapse or break down the bone matrix, so that the calcium can be released into the bloodstream. Calcium, when the levels get too high within the bloodstream, which is not good either, calcitonin is released from the thyroid and inhibits osteoclasts, so it reduces the release of calcium and increases absorption of calcium. This is, as I said, a type of endocrine signaling. So, what are some important things to remember about the immune system? What is the difference between a pathogen and an antigen? So, a pathogen is any substance that isn't supposed to be within your body. So it can be a fungus, a virus, a bacteria, a protist, anything like that. But what is an antigen? An antigen is actually a protein that is found within the outer membrane of a pathogen. So, what is a PAMP? A PAMP is a pathogen-associated molecular pattern. It's basically a molecular pattern or molecular shape that we do not find within our own bodies. So, a couple other reminders about the immune system. White blood cells are all referred to as leukocytes, while red blood cells are called erythrocytes. And then we go on to the lymphocytes. Most of the time, people call them B cells or T cells. And we'll talk a little bit more about B cells and T cells in just a moment. But basically, they are both lymphocytes, and they are born in the red marrow cavities of the bone. That's why we came from the bone over to the immune system. What's very important is a B cell will mature within the bone, where a T cell will mature within the thyroid. So let's take a look. The three-step line of defense is one of the most important things to know about the immune system. So our first line of defense includes surface barriers. This could be a skin or integument of any kind, mucous membranes or different secretions, oils on your skin, the pH of your tears and your saliva, anything like that can work as a surface barrier, and that should be considered your first line of defense. Then we have innate immunity. The innate immunity is what we are all born with, and we didn't have to learn anything else. And this includes a lot of phagocytic cells, especially neutrophils and macrophages. Natural killer cells are another type. Antimicrobial proteins go along with innate immunity, and they are considered to be complements and interferons. And there's complements and interferons basically are really important for interferons basically inhibit viral proteins from being formed, and then complements are really just circulating inside of the bloodstream, and they don't do anything until they encounter a PAMP. Inflammatory response is a response that initiates heat and inflammation, allowing a little bit of swelling and allowing some of your white blood cells to get into areas they normally could not. Our third one is adaptive immunity. We have humoral response, and your humors are the liquids of your body. Antibodies from B cells patrol those body fluids. And then we have cell-mediated response, which would include something like cytotoxic T cells, which defend against infected body cells. So let's look a little bit more at B cells and T cells, shall we? So B cells, when they encounter some information about a foreign body or an antigen inside of your body, they are going to initiate a response. Part of their response is to clone themselves, and B cells can produce plasma cells, which are really, really important, and they're going to have memory cells. So a B cell creates some plasma cells, and what's amazing about this clonal response is the antibodies that are all over the outside of those cells are able to be released and float through all of the liquids within the body. So these antibodies are really, really important. They are going to be released. They're going to go over to some antigen. They're going to bind to the outside of it. They're going to keep it from binding to other cells, and they're pretty much going to mark it for death. Another thing that's important is that a B cell can help a T cell learn to recognize this foreign particle. So T cells will often bond to an antigen presenting cell, and an antigen presenting cell is a cell that has become infected. It has taken parts of that infectious particle, it's chopped it all up, and it is presenting it on the outside of its cell membrane on these things called MHC markers. MHC markers are major histocompatibility markers, but one of the things that's important about them is they help us determine self versus non-self. So kind of an important concept. All right, let's continue forward. There we go. Now let's talk a little bit, just a little bit, about the endocrine system. So the endocrine glands are some of the things that create and distribute the hormones that we talk about when we talk about endocrine signaling. We've talked about a few of the different ones. We could talk about the thyroid, the parathyroid, the adrenal cortex, the kidneys. We can talk about a lot of the different ones. Some of these are considered to be steroid hormones, but some are still considered to be a peptide or a protein hormone. Now let's talk about long distance endocrine signaling. And one of the most important, in my opinion, examples you can know of homeostatic control is actually blood glucose regulation, which is a negative feedback control. When your blood sugar is high, your pancreas stimulates insulin, which is going to tell your liver to release glucose. Or if it is within the blood sugar itself, it is going to have insulin that is going to be released, which is going to act as a signaling molecule for your tissue cells to uptake the blood sugar or the food, if you will, from the bloodstream. Now, if your blood sugar is too low, the pancreas is going to release glucagon, which is going to initiate the stored glycogen to be broken down into the monomers of glucose so that it can be utilized in the bloodstream to feed your cells. Whew, that was a challenge for me to get through for some reason today. One of the other hormones that I wanted to talk to you about was ADH, or antidiuretic hormone, which is released from the posterior pituitary gland. It is going to be very important in helping to regulate the amount of water within the bloodstream. It is going to either dilute your urine or concentrate your urine based on several other factors, but ADH in different situations can have um, different results according to which other hormones and signaling molecules are signaled after the ADH. Now let's talk about some plant hormones. So some plant hormones to think about include auxin. Auxin is really an important one. There's a lot of different varieties, including one that is called indolbutyric acid, and sometimes they call that IBA, but it has several other names as well. It is one of the main controlling hormones or plant growth regulators, like they like to say, within the um, plant regulation system. Cytokinins is another plant hormone or plant growth regulator, and it is going to aid in cell division and differentiation. But guess what? It doesn't ever work alone. It works with auxin. Um, uh, Gibberellins is how I say the next one. Gibberellins is kind of an interesting one. It has the ability for stem elongation, but also for fruit growth and seed germination. Gibberellins are the reason that we have these Thompson seedless grapes that are really, really big and long because they have been treated with gibberellins. Racinosteroids is another one. It has to do with cell elongation and division in the stems. Then we have abscisic acid or ABA. Interestingly, they used to think that one had to do with abscission, which is the aging and falling off of leaves, but they found out later on that it did not. One of the most amazing ones, in my opinion, is ethylene, and ethylene is a gas that is released. It's heavily involved in signaling for mainly fruit ripening, but it also can be as a response to a predator. So let's look at one of the experiments that was done where they wanted to look at phototrophism within little shoots of plants. So they did some experiments, and in 1880, it was Darwin and his son Darwin, and they decided they would do these different seedlings in a variety of different treatments. The center diagram shows it the best, where the tip of it, called the coleoptile was removed. Then they took and covered it in the next one with an opaque cap so that light could not get through. Then the next one, they covered it, but it was with a transparent cap so that light could get through. And then they took the base of it and covered it with an opaque shield so that light couldn't get to that area. They didn't really know what was causing that bending towards the light. And they found out that it was actually the hormones that were produced inside of the tip of the root. Of course, auxin was involved, and that allowed for a cell wall to become, the extracellular matrix of the cell wall to become more pliable, and so that the outer area, the one away from the sun, that could stretch and elongate the extracellular matrix, which we would call the cell wall. Interestingly enough, that wasn't enough. Of course, in 1913, the Boysen-Jensen experiment decided they would do something kind of similar to make sure it really was coming from the tip, the signaling molecules were. So they cut the tip off of two different seedlings, and then they placed a little cube of agar on one, which was permeable to the auxin, and then they did mica, which is a mineral that's very glass-like, and they put that in between, and it would not allow for the auxin to go through. So they did figure out that the tip is where this hormone was being produced. All right, let's talk about the triple response just a little bit because it's funny and cool. And the triple response is due to some type of a mechanical stress on a plant. Like if you had some tulips and they needed to come up from the ground and you had accidentally put a pot right on top of where those tulips were planted, they would come up, they would feel a barrier. They would stop growing forward. The stem would thicken and then it would start to jut out to the side before it would actually go back up again. That's known as the triple response. It's like stop, bend, twist. It's kind of an interesting response to some environmental stresses. I really hope that this was helpful. I made a lot of mistakes and had some challenges along the way, and I have to do a lot of editing. So you have a great night. Good luck studying.